Welcome to Brain Ponderings. I'm the host, Mark Matson, and Happy New Year to everybody. Today, my guest is Professor Keqiang. He has two places, actually. He goes back and forth now between Emory University in Atlanta and China in a place just south of Hong Kong called Shenzhen. And in his background is he grew up in China and he did his bachelor's work in chemistry there, then master's in chemistry. Then he came to Atlanta, to the United States, to do work at Emory University, where he got his PhD. And looking at his CV, it was in things related to cancer and cell death. And now he's very interested in neurodegenerative disorders that, of course, Alzheimer's, Parkinson's, for example, that involve cell death. And so, welcome.
Thank you very much, Professor Madsen, for your invitation. It's really my great pleasure to have this wonderful opportunity to meet you face to face. Though I have read a lot of your paper, but it's the first time to see you on the screen. And though we have know each other from the literature, and I also know many of your previous trainees, but it's really my great pleasure to have this wonderful opportunity to talking to you face to face.
I'm looking forward to this. The main focus of the talk will be on your work on looking at gut-brain connections in neurodegenerative disorders, and in particular, how things going on in the gut and neurons innervating the gut may uh, be early in Alzheimer's, Parkinson's, and how certain proteins there that you'll talk about may propagate disease back up to the brain. So can you give a little bit of background? So you grew up in China. What was that like? Were any of your parents, either of your parents, scientists?
No, actually, now my parents are scientists. Uh, my mom is a farmer. My father is a worker. But since I was a kid, I was very interested in science, especially chemistry. So when I was in high school and in college, I want to be a chemist. So that's why I chosen the top university in China, which is well known for its chemistry. That's why I chosen to go to Jinin University, which is in northeast of China. After I graduated from Jinin University, I went to Beijing University, which is also well known for its chemistry. So I got my master's over there in 1990. Then I chosen to join Emory as a graduate student. At the beginning, I was also in chemistry department to study organic chemistry.
Ah,
That's why even now I'm using the chemistry as a tool to do a lot of drug discovery and modifications. So then after 1.5 years in chemistry department at Emory, then I transferred to biochemistry because I realized it's more interesting and more disease oriented in biochemistry. That's why I shift to from chemistry department to cell biology department and working on cancer. Because during my rotation, I made a very surprising discovery. I discovered a nature product called anoscopine, which is opioid alkaloid. And because Saul Snyder also working with this opium alkaloid before, oh, excuse me, I have a visit. It's surprising. Sorry.
okay. So Dr. Ye, so as you heard, he's trained in chemistry and biochemistry, and he started working on cell death pathways and natural products, so molecules produced by plants uh, that
I'll come back.
affect cells. And okay, so then you got a appreciation and background in cell biology, right? Because you're studying cell death. So this is a good way to go actually from chemistry to biochemistry, then to cell biology, because you're going kind of from the basics and then on up to increasing levels of complexity and how you get interactions and how they're modified by the environment, which you'll talk about, the gut, the microbiota, for example. And
Exactly.
okay, so then after you got that training and kind of combining your expertise in chemistry, biochemistry with cell
Yeah.
biology, so
Uh-huh.
you were in Baltimore, where I am now at Johns Hopkins University, with Saul Snyder, who's one of the most highly, I think he's the most highly cited
Yes.
neuroscientist in the world. Like
Yeah.
last time I checked, like 1,200
Yeah.
publications.
Yeah.
He retired recently.
Yeah. Mm.
What was his
Last year.
lab? But
Yeah.
he's a
Mm.
wonderful guy.
Mm.
And so there, you were studying. So Saul played a major role in discovery of opioid receptors long time ago.
Yeah.
And then he's done a lot of work. He got interested in his background is in psychiatry, but he got very interested in neurodegenerative disorders uh, towards the last part of his career. And what did you study with him? These
Yeah. When I joined his lab,
lipid
yeah.
derived molecules? Or,
Uh,
no?
yes. I working on a protein called PIKE, PI3 kinase enhancer, which I discovered using East2 hybrid system screening when I joined his lab. And then we discovered this very interesting small GTPase. It can binds to PI3 kinase and enhance PI3 kinase activity, mediating neurotrophic factor from the plasma membrane to the nucleus.
Ah,
So,
yeah.
yeah, and this GTPase play essential role to coupling those neurotrophic factor to PI3 kinase AKT signaling. If we knock out or delete this GTPase, then NGF, BDNF will fail to activate PI3 kinase activity.
Did you
So
look at so the receptors for the neurotrophic, some neurotrophic factors are a couple of this pathway, but also insulin, right? So the insulin signaling pathway, insulin binds to its receptor, the same, or one of those, signals to transcription factors and then nucleus and then affects gene expression.
yeah. So mainly we focus on BDNF, NGF, and we found track A, track B receptor using this GTPase to activate PI3 kinase, passing the pro-survival signaling from cytoplasma membrane to the nucleus. So I published this study in Cell and Nature and during that time. So when I get my faculty position, Saul was very generous to allow me to continue working on this protein. So I carry on this project, working on this protein called a PIKE, PI3 kinase enhancer. Because I work on this very interesting GTPase later on, that's why I discovered the AEP, asparagine endopeptidase. This is the enzyme. Now I spend in the past 15 years, my lab focused on dissecting this protease role in driving Alzheimer's disease and Parkinson disease. So it was a serendipity when I discovered this AEP because we prepare a wrong buffer. So it was solely serendipity because Pike main function is pro-survival. So we did a product assay find a nuclear protein called SET, S-E-T, which is a small protein in the nucleus the main function for SET is act as a DNase inhibitor. It can bind to DNase and inhibit the DNase activity. However, when a SET is caught by AEP, it will lose the anti-DNase activity, and it can, by the way, it also has a lot of function to block PP2A to prevent tau hyperphosphorylation. So once SET is cleaved by AEP, it will lose its inhibitory activity against the PP2A, leading to tau hyperphosphorylation. So when we try to dissect how
And Keqiang,
yeah.
this would be a good time to give some background on for people who may not be aware of the relation to Alzheimer's, Parkinson's, for example. Can you talk about the proteins that are implicated strongly in those diseases in terms of cell death?
Yeah, okay.
And then the enzymes you discovered that cleave those proteins?
Yeah. So this enzyme, because we discovered this enzyme at the beginning, we did not realize what is the pathological role or even physiological role, because we discovered it solely by serendipity due to the wrong buffer. So if we're supposed to prepare a pH 7.4 at a neutral buffer, accidentally we prepare acidic
Ah,
buffer. But this enzyme is a lysosome enzyme. Its
ah,
activation require acidic pH, such as 5 or 6 pH. So at that time, we're using the stroke model to dissect the role of AEP in ischemia, because under the stroke condition, the brain pH change into acidic. That's how AEP is activated. It can cut a set and leading to DNase activation, leading
ah,
to a neuronal cell death.
ah.
Then we ask, besides stroke, any other condition our brain's pH can drop to acidic. So we through the search the literature, interesting, we found out, oh, there's some brain region, even during the aging process, when we're getting old, some specific brain region pH could drop from 7.2 or 3 to 6.8, 6.9. So we are wondering what is possible in those brain regions AEP could be activated, and what is the main disease associated with the brain aging? Obvious, Alzheimer's disease is the well-known disease associated with brain aging. So that's why we test whether it's possible this protease could cut the well-known major player in Alzheimer's disease, such as APP and tau. And APP is a membrane protein, is a amyloid precursor protein. And the previous study have demonstrated it can be cut by alpha, beta, or gamma secretase to mediate A beta production, the metabolism of this membrane protein. So on the other hand, tau can also be cut or hyperphosphorylated and form aggregate within the neuron. On the other hand, the A beta secreted form cleavage of APP by base or gamma secretase cleavage, it can deposit extra cellularly. So then we next test whether it's possible AEP could directly cut this major player and play some role in mediate the Alzheimer's disease pathogenesis. So that's where we start our discovery. So we published this paper in 2014 in Nature Medicine and 2015 in Nature Communication, reporting AEP could function as a delta secretase because it can cut APP mainly at N585. By the way, AEP is estrogen endopeptidase. It's a cysteine protease. It belongs to the same family member of caspase, but
Mm.
AEP only activated under acidic condition, caspase activated under the neutral pH.
And
So
where in the cell is AAP? Is it in the lysosomes? Is it in the cytosol? Is it in the nucleus? It is in all those components?
AEP mainly resident within the endolysosome. So the receptor, for example, APP, this transmembrane protein, after its endocytosis, it can merge from earlier endosome to a later endosome where it meet with the
I see.
lysosome and with the AEP, where it can be shredded by AEP. So the cleavage site is about 11 amino acid away from beta secretase cutting site
Okay.
because beta secretase cut at N596 uh, and AEP cut at N585, so only 11 amino acid away. So we previous study disclosed this cleavage is the prerequisite for beta secretase to cut APP. And on the other hand, we also reported AEP could direct cut beta secretase to enhance its enzymatic activity, to enhance the secretase activity. In the other word, delta secretase, AEP, could simultaneously cut the substrate and also can cut the upstream beta secretase to facilitate A beta production. So interesting, this enzyme not only cut APP mediate A beta production, it can also cut tau flanking microtubule binding site. One is N585, one is N386. Those two sites just flanking the four repeat domains to stimulate tau hyper
Huh.
aggregation and phosphorylation. In our previous Nature Medicine paper, we find out it is this cleavage take place before the hyperphosphorylation because we find out the cleavage can enhance phosphorylation, but phosphorylation does not regulate this cleavage. So if we block the cleavage, then the phosphorylation will substantially decreased. So we iron out the molecular sequence. We find out is this cleavage take place first, then the hyperphosphorylation, then the aggregation of tau within the neuron. So,
A few comments there. We did some work a long time ago, actually when I was at University of Kentucky, uh, trying to understand the normal function of APP, which there may be more than one. But
yeah.
one enzyme you didn't mention is called alpha secretase, and it cleaves kind of in the middle of the amyloid beta peptide sequence.
Yeah,
So the alpha secretase cleavage will prevent A beta
production.
production.
Yes.
And we did some work suggesting that the product generated by the alpha secretase, secreted APP alpha, plays important roles in regulating neuronal excitability,
Mm-hmm.
synaptic plasticity. I actually had a postdoc, Katsutoshi Furukawa, who got a Nature paper out of that.
Yeah, the P3 peptide, right?
Yeah.
Yeah, yeah.
And
Mediate
then,
the synaptic
my point
plasticity.
there is there's a normal function of APP and there's work going on. It may be important for,
Yes, there is a balance between the amyloid genesis and non-amyloid genesis.
yeah. And then tau, it's normal. You mentioned it binds the microtubule. Its normal function is kind of stabilize the microtubules, which in neurons they have long axons, and it's important. Obviously, it's important to maintain the structure of axons and dendrites too, and to have the ability of those processes to change structurally. So
Exactly.
if tau comes off the microtubules, that's not good in itself.
Yeah, for the axon dynamics and for the
Yeah,
axon to function as a track for train track for the
yeah.
cargo to transport.
A lot of things.
On-
Mitochondria move along them. We've
yes,
had podcasts
exactly.
on all those things before.
Yeah.
Okay, so this AEP then, the way you're describing it, it's important in generating, if you will, pathogenic A beta and tau,
Hmm.
self-aggregating forms of these molecules. And so in theory then, a drug or another approach that inhibits AEP should have a beneficial effect in Alzheimer's? Whoop. Okay, right. So you mentioned before we went on that you're involved with a company
In
in China.
China, I
Actually,
found a company called Bridgen Pharmaceutical, Bregen. It stands for brain in Greek. So
ah,
I found this company, and the main business is working on a few products which I developed at Emory University. So this company licensed four patents from Emory University, which they all come from my own laboratory. One is the tracklist, one is the AEP inhibitor, and the other one is the most recent product we discovered is called alpha-synuclein PET tracer.
ah, okay, and that's the one protein with. So you've talked about Alzheimer's disease and your discovery of this enzyme that's plays a
Are you
very
recording?
important role in
Looks like
cleaving.
the internet was not quite stable.
No, I'm not sure which end it's on, but actually your voice is still fine. So
Oh, okay.
that's the main thing. Um,
Yes.
okay, so then what about Parkinson's disease? You mentioned you're developing a way to image a protein called alpha-synuclein that
Yes.
abnormally accumulates in neurons. That is to image in living, well, you've done it in living mice, but the idea is to use this in humans too as kind of a way, what could be for
Mark,
diagnostics.
can
It
you hear
could,
me?
yeah, I can hear you.
Hmm.
Yeah, I can hear you. It's fine.
Oh, okay. So let's continue. So because alpha-synuclein in Parkinson disease play an essential role and its function kind of like tau in Alzheimer's disease. So we reasoned if AEP could cut tau and facilitate this pathology in Alzheimer's disease, whether it's possible AEP could cut alpha-synuclein and mediate its role in Parkinson's disease. So then we just extend our study from AD into the PD. So interesting, in 2017, we published a Nature Structure and Molecular Biology paper reporting AEP could cut human alpha-synuclein at N103. And after this cleavage, it can enhance alpha-synuclein aggregation. And moreover, we found this cleaved fragment in Parkinson's disease patient CSF and also in their brain samples. And also, if we inject this fiber after cleavage, and it prone to form fiber much faster compared to fullness alpha-synuclein. And this truncated fiber is much more toxic to driven uh, dopaminergic neurodegeneration. And if we inject this fiber into the brain, its propagation is much faster compared to the fullness uh, alpha-synuclein formed aggregates. So then we ask uh, why AEP plays so important role in both neurodegenerative disease, whether this protein is age-dependent um, regulated or not. So this leading to our discovery of a crucial transcription factor, CEBP beta. CEBP beta is very interesting transcription factor. The major physiological function of this transcription factor is to mediate adipocyte differentiation by mediate a lot of fat related metabolism enzymes synthesize. And moreover, it has another function mediate the inflammatory cytokine expression, CB beta. So we find out it can dictate AEP expression in neuron and also in astrocyte and microglia in age-dependent manner. Besides, AEP is mediated by CB beta. Interestingly, we find out one of the key major player in Alzheimer's disease and Parkinson disease are also regulated by this transcription factor. For example, ApoE4 and APP MAPT, which is a gene name for tau, and also alpha-synuclein and beta secretase. I just gave you a short list. One of these major player are all the substrate downstream target of CB beta. Oh, the other thing I forgot to mention is MAO-B, monoamine oxidase, which is one of the key enzyme to metabolize dopamine into dopal and other metabolite. Because of Parkinson disease, there's some inhibitors for MAO-B inhibitors can be used for treating Parkinson disease.
Oh,
So
okay. So the AAP, uh, the gene encoding AAP is regulated by this transcription factor CB
CEBP beta,
beta. And then that transcription factor is also regulating the genes encoding these disease-related proteins.
exactly.
Now, have knockout mice been do if you knock out the gene for AAP, do mice survive?
Yes, and those has been well studied. And VEP was original clone from the plant called a legumin. It's a vacuolar processing enzyme, mainly mediate the plant agent. For example, AEP in the plant, it is a mediate the leaf color from green into yellow during the transition from the summer to the fall season. So
Interesting.
yeah, it's a very original clone identified in the plant. It play critical role, mediate the life cycle.
Is there a pH change in the leaves as they turn from green to yellow or red?
Exactly. Yes, the vacuolar membrane is getting thinner, and then the VPE, called a VEP in mammalian cells in the plant, is called a VPE or legumin. So it can leaking out with the pH change. It can cut the
So
grain.
that's similar to having a lysosome become dysfunctional and leak out. Uh, I don't know. You know the chemistry more than me, but obviously the pH is based on hydrogen ion.
Yes,
So
exactly.
they get a change in hydrogen ion concentration around the lysosome or something like that.
Yes, so actually, EEP in uh, uh, vertebrate, it can implicate some, uh, for example, human diseases. One of them is we published in the knockout mice in 2010 in PNS. We report it can engulf red blood cell and mediate the immune function. And also AEP knockout mice has also been reported implicate in kidney functions because usually AEP is highly expressed in our kidney and mediate the recycling of the proteins. So knockout mice, it can survive, no problem. And the heterozygous mice, just like 100% normal, kind of like a beta secretase knockout mice is normal. So if we cross the AD mice or PD mice with AEP knockout mice, we can substantially block the AD or PD pathologies. So indicating this enzyme might be a good drug target. So that's why we perform a high throughput screening. We identify a small molecule. Now it's in IND stage. So we are doing the IND enabling study and maybe file the FDA application soon to initiate a clinical trial. And also Roche, the big pharma, is
Yeah.
also targeting this protease for Alzheimer's disease. They have published several papers about their drug development programs, discoveries. Yeah. So
Now,
that's
one thing in developing that kind of drug, so for the brain, we'll talk about the gut pretty soon now, but
yeah.
uh, one issue in developing drugs for the brain is the blood-brain barrier, and do they cross? And so, yeah, how do you approach that? I guess you want something that's like lipophilic that but still interacts with the enzyme. Is that the idea?
Mhm. Yeah. So certainly, because our main target is the neurodegenerative disease, a small molecule passing the BBB, which will be the major priority we always keep in our mind. So doing the drug screening after we identify a small, short list of the candidate, we put the brain permeability, brain exposure on the top of
Yeah.
our priority.
Yeah.
So then we analyze those small molecules in vitro ADMET called absorption, uh, uh, distribution, metabolism, and excretion and toxicity. Doing those PK profiling, then we only focus on those small molecules that demonstrate good lipophilic activity, demonstrate good um, brain permeability. So those will
So
be
are
lead
you
compound.
screening? Are you screening? So you did initially like a big screen. There's these we call libraries of chemicals,
Yes.
and a lot of those chemicals are maybe I guess I don't know for sure. A lot of those chemicals that maybe you screened, are they plant products? Do you think there's
The
natural small molecule
yeah.
inhibitors of the enzyme?
Yeah, it's up to the scientists to choose which library you will use. And we use a very big library. We published this work in 2017 in Nature Communication, and we screen about 55,000 compounds library, which contain natural product, contain FDA-approved drug, also contain the druggable top candidates, and those library are commercially available. So we choose those library which has a higher druggability, and because they meet the Lipinski rule of five and have
I
a
see.
good lipophilic activity. So at the beginning, we intentionally to choose those kind of library to looking for our candidate. So fortunately, we have identified lead compound. It functions as an allosteric inhibitor of AEP, and it has good oral bioavailability and has a pretty good BP ratio, the brain and the plasma ratio. So indicating this compound not only is orally bioavailable and also is brain permeable, so it can go in the brain and demonstrate good half-life. So that's why now we are doing the IND enabling, and we extend this study in from the Alzheimer's disease to Parkinson disease, and we also demonstrate that this compound now it has a very good therapeutic effect in atherosclerosis and also even in the osteoarthritis, because those are the bone degenerative disease, and it can block AEP cleavage of the MMP3, MMP13, which play critical role in osteoarthritis and
So
also,
inflammation.
yeah, inflammation. Yes,
Yeah.
exactly. Because AEP play critical role in inflammation and in many uh, lesioned tissues. Yes.
Okay, well, this will be exciting to see in the next 5 to 10 years how the human studies go. Now, of course, I'm friend with Ted and Valina Dawson. We collaborated on, well, one main study actually, but and we got interested in the gut. So go ahead and talk about the gut. This kind of originated from some histological work done by Heiko Brack,
Mm,
right? And oh, there's a couple background things. So in Parkinson's disease, clinically, many patients have chronic constipation. So that's gut problem, right?
exactly.
And then there was this evidence, I can't remember when it was published, but there people with gastric ulcers, I guess that's ulcer in the intestine. One of the treatments, if it's real severe, used to be to cut the vagus nerve, which is well, that's a nerve that innervates the gut and it promotes secretion of acid, I guess. And and then they followed these patients over decades and decades, and there was a greatly reduced incidence of Parkinson's.
Mm,
Suggesting if you cut the vagus somehow. Okay,
yes.
so pick it up from there.
Okay. So as you indicated, there's some prodromal symptoms such as constipation is a very obvious in Parkinson disease patient. So that's why we extend our study from uh, the brain into the gut, and the gut also called the second brain, and because the second organ in most abundant in the nerves. So that's why we extend our study from the head over our shoulder to the belly. So interesting, we made a similar observation because in the patient gut, first of all, we got the biopsy sample from Parkinson disease. We performed immunohistochemistry staining using our antibodies specifically recognize AEP truncated alpha-synuclein, AEP truncated tau, specifically is N103 and tau N368. So both antibody are very specific. Interesting, we find out our specific antibody could recognize the truncated fragment in Parkinson disease gut tissues. By contrast, in the healthy biopsy from some other colon diseases, we did not find the cleavage, so indicating presumably AEP is activated in the colon of the Parkinson disease patient where it can cut the upregulated tau and alpha-synuclein. Previous study has been reported reported fullness alpha-synuclein and tau, they could physically interact with each other and exacerbate their biochemical function, not only in the fiber formation and also in their propagation. So that's why we investigate whether this AEP cleavage could play some role in exacerbate or dictating alpha-synuclein and tau pathology in the Parkinson disease patient. Moreover, we also extend our study from the Parkinson disease to Alzheimer's disease. I still remember I reading your previous paper about the in the mutant fasting regulate the our brain functions and mediate our brain agent. And also, I remember in your previous study, you reported that the NAD mediate the brain functions and doing those intermittent fasting due to the calorie restrictions. So because we are what we are eating, so what are our lifestyle play an essential role, which is kind of called the environmental factor mediating cross-talking with where our genetic internal factors, the external factor communicate with internal factors and play essential role dictating the disease onset. So that's why we extend our study from PD into the AD. We were also asking whether it's possible what we discovered with the colon tissue from biopsy from the Parkinson disease also take place from the Alzheimer's disease patient. So by collaborating with a doctor in Beijing and who also obtained the Alzheimer's disease gut tissues, we performed immunostaining. We also find out APP is also truncated by AEP on the gut tissue from Alzheimer's disease patient. And we also discovered there's a lot of A beta deposit on the gut tissue as well, just kind of a small centralized brain in the gut. So that's why we extend our study from PD into the AD, and we also perform the similar experiment. We inject the Alzheimer's disease brain extract or the brain extract from Alzheimer's disease animal models into the wild-type mice colon or into the young 3TG Alzheimer's disease AD mouse models intestine muscle layer. And we can observe those aggregated fluorescent labeled A beta or tau, they could spread along vagus nerve from the gut into the brainstem, from where it can spread into the different brain regions. So now we can extend our study from the from the PD to AD and from the gut into the brain. So we now question ourselves whether Alzheimer's disease or Parkinson's disease really originated, at least for a portion of patient, is from our gut or not. Now, whether it's possible we could do earlier diagnosis of those patients before those pathogens spread along the vagus nerve from the gut into the brain. If that's the case, then we could do the earlier diagnosis at least 10 or even 20 years before those neurotoxic pathogen being spread from gut into the brain. Then, if that's the case, then I believe we could find a way to stop this disease progression and kill this disease at its earlier stage before it becomes to a neural disease. Actually, is a peripheral, is a gut disease, is a GI disease. So
Uh,
that's why,
there, there, there are rare mutations. Uh, they're inherited forms of Alzheimer's and Parkinson's, and they have early age of onset, and they can be caused, in the case of Alzheimer's, by APP mutation
and the PS1
or
mutations.
PS1, even more
Mhm.
commonly. And
Yeah.
actually, PS1, and like for example, down in South America, in Colombia, there's these families where they have these mutations that if you have the mutated gamma secretase,
Yeah,
essentially,
Churchill crest. Yeah.
yeah. And
Yeah.
so, what about could you get intestinal biopsy tissue from those people when they're younger, when
Mm.
before they're symptomatic? Because you know for sure, 100%.
They will have the AD in
I
their.
don't know because these are they're
Yeah.
generally there's kind of ethical things and cultural issues too to get the people to volunteer.
Yeah.
Uh, but
That's
how hard is it to do a intestinal biopsy? Is there any risk involved to the patient or
No, actually, for example, the for the AD-PD patient, the reason we get the biopsy because they have the complaint about their GI problems. That's why the doctor doing the physical examination to examine what's going on for their colon tissues. That's how we
Oh,
get.
of course. I just had a colonoscopy recently too, and
Yeah,
they
actually, it's quite frequent for people older than 45. A doctor recommend for us to do the routine annual examination.
yeah, mine was clean, but some people have these polyps which are kind of precancerous, and then they'll do a biopsy, take them
Yeah,
out. But
yeah,
when those biopsies are done, so there's this mesh of neurons on around the gut,
yes.
and
Mhm.
then but the first cells inside the gut are the epithelial cells.
Mm.
So when they do a biopsy, do they get neurons or just the epithelial cell?
Maybe a portion of them is epithelia, and there's some submucosal plexus which contain a small portion of the neurons where you could identify because there's a two layer of neurons in the gut. And the outside is the and the muscle, that's the muscular one, and the deeper ones, which is easy for doctor to get a biopsy,
Okay.
is the submucosal, which is right below the venar and the epithelial layer. So
I
where
see.
you could uh, have some a small portion of the submucosal plexus, which contain a lot of different neurons. Yeah, as you just mentioned, the Christchurch mutation, which is ApoE3R136S mutations, which can block the earlier PS1 mutation
Oh,
induced AD. We have a neuron paper actually come out next week. We also find out uh, this mutant actually bind to tau and stop tau spreading, which is one of the reasons why those PS1 familiar AD patient, they do not demonstrate their clinical symptom even until their later 70s. So we find out these mutations, ApoE3 mutations, can bind to tau and block tau pathology spreading. Yeah, so this paper come out next week.
that's
So
exciting. So, yeah, that's a very rare. It's a good mutation
mm,
from the perspective of Alzheimer's. It's protective.
yeah, so we are even think have testing a crazy idea. We are thinking about whether this mutation can be used for clinical application to treat Alzheimer's disease patient or not. So now we're testing in animal models. We just inject with 3xTG or 5x
Yeah.
FAD mice with this AAV encoded mutant, what is sufficient to block the AD pathology outside or not. If that's the case, then mankind will have a powerful genetic drug.
Ah, yes.
Yeah.
So you're saying, yeah, of course, technically, and it's a lot harder to inject things in the brain than in the gut. But if if you can put this good mutation in the gut cells, is that what you're doing? And if that,
We
if,
use two approaches. One is use the cell transplantation, which actually is approved by FDA used tool. And the second can use the AAV virus, and
yeah,
which is also can be
but are you doing it in the
approved by
gut or the
FDA.
brain or both?
Yeah, you can do both. We can directly inject
Yeah,
in animal. We inject into the gut and also inject into brain. Use two different approaches,
yeah.
and I'm conceivably, if this works in future, we could for the human patient, we could use the AAV virus inject into the brain, which is approved by FDA. Some of the backbone viral therapy, you can even imaginably can block the AD pathogenesis in the earlier stage, before most of neurons are degenerated, because this mutant is so powerful and amazing.
What would happen if, so the vagus nerve is very big.
We
Uh,
inject into the colon.
yeah, yeah, inject into the colon.
Mhm.
The,
Where
the colon's
the
very big.
synapse will spread.
Yeah,
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 those are reported before the viral injected protein, they can transport along the vagus nerve into the brain. Those has been reported before. Yeah.
yeah. All right, then, um, as you know, over the last 10 years or more, there's been a big proliferation of research on the gut microbiota, with most of the focus on the bacteria in the gut, although there's
Yes.
also
Yeah,
viruses in the gut.
so after we discover this pathology in the gut, the next obvious we ask, uh, what is going on for the role of gut microbiota in the ADPD patient? Whether it's possible really find the pro bio biotas could do some good things for stopping AD or PD. That's why we spend some effort to generate the germ-free AD mice. So a few years ago, we report in the gut using the germ-free mice in 3xTG. We find out the PUFA pathway is a polyunsaturated fatty acid, mainly is arachidonic acid and PGE2. They are highly upregulated in AD brain and play a critical role in driving the AD pathogenesis, because we identify one of the bacterial is Fragilis. This bacterial mainly responsible for metabolize arachidonic acid into the PGE2. It can drive the inflammation
Mhm.
and then activate a microglia. So that's why we spend a lot of effort to dissect this uh, uh, pathogens biological role in mediate the AD pathogenesis. And we also performed a similar study in Parkinson disease, because for Parkinson disease, the gut microbiota, there's an extensive studied by a lot of laboratories. However, it lacks the consensus exactly which few of the bacteria play a critical role in PD pathogenesis. So one of the approach we used is take advantage of the animal models, because in humans, there are so many things could influence
Yeah.
your microbiotas, your ethnicity, and your living style, and your diet,
Yeah.
the region you are living, or your medication, many things. So that's why we use the animal models. So we're using the alpha-synuclein transgenic mice, because these mice can faithfully mimic upregulation of alpha-synuclein. Even wild-type alpha-synuclein duplication can trigger PD pathogenesis, and also rotenone, which is one of the pesticide, has been well characterized
Right.
in humans or in PD mouse model driving PD pathogenesis. So we're using the combination of these two model in one models, because using the lower dose of rotenone and alpha-synuclein transgenic mice, it can faithfully duplicate what happened in Parkinson disease patient, because alpha-synuclein transgenic mice does not have the alpha-synuclein Lewy body formation, and it can only mimic dopaminergic degeneration or the motor impairment. That combined with rotenone, then we can reconstitute the Lewy body formation. So using this model, then now we could monitor the gut microbiota, and interestingly, we find out one of the bacterial hepaticus, this inflammation-driven bacteria play a critical role in mediate dopaminergic neuronal cell death, and also mediate alpha-synuclein aggregation. So now we are dissecting what metabolite from this specific bacteria it can drive in the
Wow.
CEBP beta and EP pathway activation in the gut and in our brain. And what if we knock out this pathway mediated toxic metabolite production, whether we can slow down the PD pathogenesis or not? That's what we are doing in our laboratory.
Yeah, I think my understanding of the different bacterial species in the gut is that some of them produce a lot of short chain fatty acids, like
Exactly.
like butyrate or propionic acid, and those are thought to be good
Yes.
bacteria. And
Mhm.
so the species and I'm looking at the title of your paper, the Bacteroides fragilis.
Mm.
Is that, have you found that it produces certain chemicals or metabolites that are bad?
Yes, for example, in Alzheimer's disease, this Fragments mainly responsible for metabolize arachidonic acid into the PGE2.
Oh, I see. Okay.
Yeah, yeah.
I see.
This one,
So it's
it's,
pro-inflammatory.
yeah, pro-inflammatory, activating the microglia and inflaming our brain and driving the PD pathogenesis, not only in the gut and also in the brain. We're also doing the similar story about uh, the Helicobacter hepaticus, which we discovered in the PD patient. And we try to dissect exactly what metabolite from this bacteria it activate the pathway we discovered in the gut. For
So do
example,
you think in that in humans, some chronic gut inflammation could be a trigger or an accelerator of the gut pathology? We did, before I retired from NIH a few years ago, I had a postdoc, Yuki Kishimoto, who we took alpha-synuclein mutant mice that develop alpha-synuclein pathology and motor dysfunction, and we caused kind of mild chronic gut inflammation by administering something called dextrane
DSS.
sulfate.
Yeah,
And
DSS.
then that exacerbated the alpha-synuclein pathology and accelerated the onset of the motor dysfunction. We didn't do a vagotomy. We didn't cut the vagus. Actually, he tried to do a lot of surgeries and wasn't easy. But he did he did look at the uh, cells in the gut and alpha-synuclein accumulation. He did look at the vagus and he did look at the brain. And he also looked at markers of inflammation, which of course they were increased in the gut and the brain, but not the blood. So that was pretty interesting.
Uh-huh. Uh, yes, so now actually you're talking about the blood. Actually, we extend our study into the blood to doing the diagnosis for the
Yeah,
Parkinson disease. Yeah, as I indicated in last year, we published a cell paper is about the PET tracer we discovered,
right.
which can recognize the aggregated alpha-synuclein in human brain. Now we are doing the clinical trial using this PET tracer to visualize, just like for A beta tau
Yeah,
in Alzheimer's disease, we're doing the same for the PD patient to visualize the Lewy body in PD patient and also extend the study from PD to DLB and MSA because those are also synucleinopathy. Those patients also contain aggregated Lewy body in different brain regions. Actually, we also extend this study from the brain to the gut.
yeah,
Yeah, a few months ago, we published a paper in Neuron. We demonstrate if we using the gut-induced alpha-synuclein fragment or tau fragment. I already explained why we're using those fragment because we identify those fragments from PD
yeah,
patient gut. And also in PD field, people usually use the use the injection of the purified man-made alpha-synuclein fiber or PD patient extract inject into the colon or intestine muscle layer. But different people using different amount and its model is not that stable. So there's a lot of variation. That's the reason we use the gut-inducible AEP truncated alpha-synuclein, AEP truncated tau fragment. And using this mice, we can, after induction, we can observe the aggregated alpha-synuclein and tau. They could spread along the vagus nerve from the colon into the brainstem and then go to a different brain region in a time-dependent manner. And moreover, we could using our PET tracer to visualize those pathology in the gut.
yeah,
So one of the reasons we're doing this is we want to extend from the animal model to human
yeah,
prodromal patient.
yeah,
That's what we are doing in China in the hospital. We have initiated, initiated clinical trial using the prodromal patient or using the clinical diagnosed Parkinson disease patient. We want to say whether we could catch up in their colon the positive signal Lewy body in the gut or not. If that's the case, what is possible, imaginable if we just doing some surgery
yeah.
to cut this colon, whether we can stop PD uh, development at a very, very earlier stage, as I indicated before, it's a neurodegenerative disease, it's a GI disease. We can do surgeries to cut it out before spread into the brain.
One thing though, doesn't, I mean, so the vagus nerve, it's a cholinergic. The neurons that innervate the gut muscle use acetylcholine, and so that promotes gut motility.
Yes.
If the vagus nerve is cut, isn't it going to make constipation worse? Or am I not thinking correctly?
We are not talking about to cut the vagus nerve. We could cut, for example, if a segment of a colon tissue contain the PET positive
Oh, I see.
Lewy body, then you just cut those
Huh.
intestines or cut those colon.
Oh, that's an interesting idea
Yeah,
that,
just like for example, if we have a colon cancer, you just
right.
cut those intestine tissues, cut those segment of the tube
Huh.
and
Yeah.
then prevent the Lewy body spreading from the belly into our brainstem. So prevent this disease change into the neurodegenerative disease. So imaginably, and then this will be revolutionize the treatment of the
Okay, so
Parkinson.
that's potential another one. You mentioned the blood though. So are you there was a neurologist that worked with me, Dimitros Papagiannis, and I
Mm,
had him do a podcast. So he's developed methods there's these small vesicles that all cells release are called exosomes or extracellular vesicles.
mm,
And Dimitros developed a method using a neuron-specific antibody. He can first isolate all these vesicles in the blood, which are coming from all sorts of cells.
mm,
He can isolate the ones that are probably coming from neurons
mm,
and then interrogate them. And he did this a lot with Alzheimer's, showing these neuron-enriched vesicles. There's increased phospho-tau and there's
mhm.
increased A beta
Yeah, actually,
42.
we are now collaborating with Kai Blino in Sweden, and we are using the AD and the PD patient blood because in the past few years, we have published 4 or 5 joint paper reported AEP activation in the blood.
Huh.
It can we can monitor or quantitatively determine uh, in the blood the AEP cleavage of APP and tau fragment. For example, tau N368 against the total ratio of tau. And this ratio is better than phospho-tau 181 or phospho-tau 217 in AD patient to predict the tau pathology and cognitive impairment. So those now has been published. So now we are doing the longitudinal study using the 80 patient blood. We try to answer how many years before an individual demonstrate the AD pathology, we could predict an individual is molecularly inflicted with AD. So because they have
Then you can treat them early on with the AAP inhibitor, for example.
exactly, that's our final goal.
And that's the way clinical trials in Alzheimer's have been mostly failures, right?
Yeah, so
Particularly with the amyloid antibodies, which I never thought was a good idea, actually.
yeah.
Uh,
Yeah. Now we have this biomarker
yeah,
using a single molecular array called the SIMOVA tool to quantitatively and very sensitively to monitor AEP cleavage of APP and tau because our previous study demonstrate AEP cleavage of APP and tau drive A beta pathology and tau pathology. And the reason we developed this quantitative tool to monitor their concentration in the blood because we want to get ready for clinical trial. We want to know our AEP inhibitor is really engage our target so that we can
yeah,
determine our clinical endpoint.
yeah.
So now we are doing the same thing for the Parkinson disease. Have developed a quantitative SIMOVA to monitor alpha-synuclein 103 in PD patient blood so that we can combine with our PET tracer for Lewy body. So then using those blood based biomarker and the brain new image, then we can monitor our AEP inhibitor whether it can shrink the Lewy body image in the brain, decrease alpha-synuclein fragment in the blood. Then we can tell the clinical endpoint for AEP. So that's the
This
whole
is all
strategy.
uh, extremely exciting work. So you've come from what you said at the beginning, your background, you you grew up on a farm? Did you
Yes,
say
in
your
the
mother was a
countryside.
farmer?
Yes.
And so you come all the way to clinical, the initial stages of getting clinical trials going in AD and PD. Just a amazing life,
Yeah,
right?
yeah. It's very exciting. Actually, when I was even a graduate student at Emory 25 years ago, the medicine I discovered, nascobin, was in phase two clinical trial, was licensed by a company in Los Angeles called Cougar Biotechnology, and they licensed the nascobin and for treating colon cancer actually, and
Oh,
it was in phase two clinical trial. I was also making a small fortune when I was a graduate student.
huh.
Yeah, yeah. The actually
What
ABC
what is
News.
the target of that drug?
Yeah, it target microtubule. It's very surprising.
Like Taxol?
It function mimic a taxol. It stabilize the microtubule,
Yeah,
prevent the cancer cell, the dynamic cell division. So
yeah.
that's how I know Solomon Snyder because Solomon Snyder studied nascobin before because nascobin is a opium alkaloid and Saul tried looking for its receptor in the brain. So I accidentally discovered this medicine when I was a graduate student, then leading to my postdoc coupling with Shaw because he studied opium alkaloid and tried looking for the receptor for nascobin. So I
Wow.
transferred myself from a cancer biologist to a neuroscientist.
Yeah. That's great. It's very exciting. Thanks for taking the time
Sure,
to
my
this.
pleasure.
I enjoyed learning what you're doing. Um,
Certainly, I want more from you because I know, as I mentioned, I know many of your previous trainees. We have some collaboration. For example, I know Ivandro Fang.
yeah.
Oh, yeah, he's working on the NAD, and actually, we are good friends. And now we have some collaboration about the NAD metabolism, some enzyme implicated in those process. We are doing some collaborations. Now he's in Oslo,
Yeah.
in Norway, right?
Yeah. Yeah.
Yeah.
Yeah. He's got a lot of things going there. They have actually, I give one lecture and they do a course on the
But the
neurobiology of
logo,
aging.
NOGO.
Yeah. Yeah.
Yeah.
Aging. Um, they're very good at aging there.
Yeah. Yeah.
Norway, they live a long time. Actually, Chinese live a long time too, I think, right?
Yeah. Yes. Many of
Better than the
countryside
United
Chinese.
States. I think one of the things that's driving the increase in AAP and this transcription factor upstream of that is obesity, diabetes, maybe even lack of exercise.
Yeah. Yeah. Hmm.
You know,
Mm.
you have any plans to do any interventions like that in your animal models?
Yes, actually now we there's a one paper under review is reporting the TrkB receptor agonist and can elongate the lifespan and by repressing
Huh,
CB beta AEP pathway. And we
huh,
have also made a similar discovery using the AEP inhibitors because 2 years ago we published a Science Advances paper demonstrate the CB beta AEP pathway upregulation in neuron can shrink or uh, shorten the lifespan in C. elegans in mice and also we have find out in human brain as well. That's why we make this animal models. If we upregulate CB beta or AEP in neuron, the mice lifespan will be shrink. So then we're thinking about what if we block
Yeah,
this
yeah,
pathway?
yeah.
So that's why we're using the AEP inhibitor. We find out in wild type mice, we can increase the lifespan about 25% from 25 to 26 month old to 33 or 34 for
That's a
wild
good
type
one.
mice. And because I'm very interested. That's why I told you AEP was originally discovered in the plant mediate uh, the plant from the green into the yellow.
Yeah.
So that's why we propose CB beta AEP pathway is the major signaling pathway driving the agent.
So
And
maybe fasting and exercise will suppress.
CB beta
Yeah,
AEP
yeah,
pathway.
yeah,
Yes.
probably.
Actually, we have demonstrated those if you are doing the exercise and also the IF intermittent fasting
Yeah,
could
yeah.
also repress the CB beta AEP pathway. That's why
Okay.
I'm reading a lot of your previous work.
Okay. Yeah. Did you get my book?
No, oh, that book I did not know it. I
Yeah.
read
After I
the
retired, I wrote a book on the science of intermittent fasting.
Oh, okay,
Yeah.
great. I know you did some clinical trial about
Yeah.
the meat and the fasting in humans and
Recently,
the
we had a
beneficial
cell
effect.
metabolism paper.
Oh, yeah, yeah,
Yeah,
yeah.
showing it had beneficial effects on cognition in older people with obesity.
Mm, okay.
Yeah.
So whether the patient who has the, let's say, metabolic syndrome could using this IF or change their lifestyle can really do the
Yeah.
therapeutic
Yeah.
effect for
Yeah, it can help. It can help even then.
Oh, okay,
Yeah.
good. I hope FDA could recommend or
Well, yeah, I hope that yeah, the government could,
yeah, yeah, could
because they
recommend.
recommend exercise,
No,
but we really need the whole. I've given a couple of videos recently on problems with the healthcare system in the United States.
mm,
It's all for profit,
mm,
and it essentially encourages people to get sick
mm.
and then treat them. And there's no preventative medicine really,
Yes. Hmm.
you know.
Yeah. So then IF will be a good lifestyle change and demonstrate a beneficial effect for many of those chronic diseases. That will be a revolutionize the treatment field and to make a big impact for
Actually,
people's
there's
life.
a lot of trials going on in cancer now
Oh, good.
because
The
the idea
IF.
is if you do fasting during chemo or radiation therapy, it will enhance the killing of the cancer cells, and it will also at the same time protect the normal cells.
Wow.
Yeah, that's been shown in a lot of animal studies. So that
Wow.
if
Uh-huh.
you go on
Thank you for letting me
clinicaltrials.gov,
know this. Mm.
yeah.
Okay. It's
Okay.
wonderful to talking to you.
Well, enjoy the rest of the time in Atlanta. Did you get any snow?
Not
No.
yet. Not
Yeah,
yet.
it's all, we got a lot of snow on the ground here. So,
Oh, really?
yeah.
Now?
No, yep. We had 6 inches yesterday.
Oh, okay. Fortunately, I will go to Baltimore at the end of this month to go to visit Saul Snyder. If possible, I will go to see you. Now you are living in Baltimore or
Yeah,
where?
we live about 20 miles north of Baltimore, Bel Air.
Oh, Belleville. Okay.
It's off 95, I-95. So it's like 25-minute drive. My lab at the NIA was on the Hopkins Bayview campus.
Uh-huh. Yeah, I know where
And
it
so
is.
it's actually that's
Mm.
right off 95. So it would take me less than a half hour to get to the lab.
Oh,
And
I
then
see.
we
Now you
have
are retired, right?
yeah.
Okay.
I still te I teach 2 courses for the Hopkins Neuroscience graduate programs one on the brain bioenergetics
Oh,
and
is that so?
the other on neurobiology of aging.
I see.
And but we have 2.5 acres, and we have a garden. We have grandchildren now,
Okay, I see.
and so I'm doing this podcast. I'm writing a little bit, and yeah,
Wonderful.
yeah,
What an enjoyable retire life.
yeah. I try to keep my brain active, but still not have all the kind of constant barrage of people coming into the office and all that.
Uh-huh.
Yeah,
As you indicated, it's an intellectual challenging. It's a good thing to stop brain aging.
yeah, that's right.
Intellectual challenging, yeah.
Yeah. Okay, Keqiang.
Okay, it's really nice talking to you. Thank you so much.
Okay, goodbye.
Okay, bye bye. Okay.